Hello everyone. Today we will discuss the topic mutagenesis and replications fidelity. Mutations and alterations in the genetic material of a cell of the living organisms or virus that is more or less permanent and that can be transmitted to the cells or the virus descendants. Mutations in the DNA of a body cells or multicellular organisms may be called the somatic mutations and may be transmitted to the descendant cells by the DNA replications and hence result in the patches of the cells having the abnormal functionality, example the cancer. The term mutations derived from the Latin term means the mutere and it meaning the change. It is an alterations in the genetic material means the genomes of the cells of the living organisms which may be virus and it is more or less permanent and can be transmitted to the cells. Mutations generally occurs in two ways, spontaneous mutations and induced mutations. Spontaneous is arise occasionally in all the cells, whereas the induced mutations is the results of the exposure to the mutagens. Spontaneous mutations. It results from the errors in the DNA replications or from the actions of the mobile genetic elements such as the transposons. Replications occurs when the nitrogenous base template nucleotide takes a rare tautomeric form, and tautomers are the isomers of a compound which differ only in the positions of the proton and the electrons, means structural positions are the different but the same. The carbon skeleton of the compound remain unchanged in spontaneous mutations, a reaction which involves the simple proton transfer in intramolecular fashions is called the tautomerism. The tautomerism means the relationships between the two structural isomers that are chemically or readily change into one another. Nitrogenous bases typically exist in the keto form and they can at times into amino and the enol forms or the immunoforms. These tautomeric shifts change the hydrogen bonding characteristics of the base and allow the purine for purine, pyrimidines for pyrimidine substitutions that can be eventually lead to stable alterations. Such substitutions known as the transitions mutations, whereas the transversion mutations where a purine and substituted for a pyrimidine and pyrimidine for a purine. In this picture, you can see the tautomerizations which I have explained in the previous lectures, previous slides. The mutations has a consensus tautomerizations during the replications and the temporary enolizations explain the formations of the 80 base pairs in the mutant and GC to 80 translations mutations occurs. The replications errors can also result in the insertions and deletions of the nucleotides. These mutations generally occur where there is a short stretch of the repeated nucleotides. In such a locations, the pairing of the templates and new strands can be displaced by the repeat sequence which lead to insertions or the deletions in the strand. Now the induced mutations. Virtually any agents which damage the DNA, alters its chemistry, or in some way interfere with its functioning and will induce the mutations. The mutations can be conveniently classified according to their mode of actions. Three common types of chemical mutagens are the base analogs, DNA modifying agents, and intercalating agents. A number of physical agents are mutagens that DNA base analogs are structurally similar to normal nitrogenous bases and can be incorporated into the growing polynucleotide chain during the replication process. These compounds typically exhibit base pairing rules, properties different from one another, and talk to replace and can eventually cause stable mutations. A widely used base analog is the 5-bromouracil and analogs of the thymine. It undergoes the tautomeric shift from the normal keto form to the enol forms and have the resemblance with the another bases. The enol tautomer forms the hydrogen bond like the cytosine pairing with the guanine rather than adenine. The mechanism of the actions of the other bases analog is similar to that of the 5-bromouracil. Now the DNA modifying agents. There are DNA modifying agents, the mutagens that changes a base's structures and therefore alter its base pair characteristics. Some of these mutagens are selective. They preferentially react with the certain bases. For example, the methyl nitrosoguanidine is alkylating agents that adds the methyl group to guanine and causing the mismatch of thymines. This is the picture in which the methyl nitrosoguanidine, this is a mutagenesis, and due to the methylations of the guanine, it becomes inserted in the structures and later on it repaired. A subsequent round of applications can then result in the GC80 transitions. Hydroxylamine is another example of the DNA modifying the agents, and it hydroxylates the C4 nitrogen of the cytosine and causing it to base pair like the thymine. Now the intercalating agents. Intercalating agents, these are the distort DNA and induce the single nucleotide pair insertions or deletions. These mutagens are planar and insert themselves between the stacked bases of the helix. This results in a mutations and possibly through the formations of a loop in the DNA. These agents include the acridines such as proflavin and acridine storage. The mutagens and the carcinogens. Many mutagens and carcinogens damage the bases so severely that hydrogen bonding between the base bases is impaired or prevented, and the DNA damaged can be no longer. And for example, the UV radiation generates the cyclobutane dimers, usually thymine dimers between the adjacent pyrimidines. Other examples are the ionizing relationships and carcinogens such as the fungal toxins, aflatoxin V1 and other benzopyrene derivatives. Now the effects of the mutations. The effect of mutation can be described on the protein label and in terms of the observed phenotype. In general, the more prevalent forms of the gene and its associated phenotype is called the wild type. Mutations from the wild type to a mutant type called the forward mutations, and it can be reversed by second mutations. When the second mutations is at the same site as the original mutations, then the reversal mutations occur. A Q reversion converts the mutant nucleotide sequence back to the wild type sequences. The suppression mutations may be within the same gene. The point mutations are the most common type of the mutations, and point mutations is protein coding genes that can affect the protein's structures in the different ways. The most common type of the point mutations are the silent mutations, in which the nonsense and difference mutations occur. Now the silent mutations. The changes in the nucleotide sequence of the codon but do not change in the amino acids encoded by the codons. This is possibly because of the degeneracy of the genetic code. Therefore, when there is a more than one codons for a given amino acid, a single base substitutions may result in the formations of the new codons for the same amino acids. For example, if the CGU its were assigned to the CGC, it would still code for the arginine. When there is a no change in the protein, then there is a no change in both the molecules. Now the missense mutations. Missense mutations involve a single base substitutions and change 8 codons for one amino acids into another. For example, the codon GAG which specifies the glutamic acid could be changed to GOG. And the effects of missense mutations vary. They alter protein structures, but the effect of these changes may range from complete loss of activity. This is because the effect of the missense mutations on the protein functions depends on the type and locations of the amino acid substitutions. Missense mutations play an important role in providing the new varieties evolutions because they often are not lethal and therefore remain in the genes. Now the nonsense mutation convert the sense codations, that is the one that code for the amino acid, to a nonsense codon, means stop codon, one that does not code for an amino acid. This causes early terminations or translations and therefore results in a shortened polypeptides. Depending on the locations of the mutations in the gene, the phenotype may be the more or less severely affected. The most proteins retains the some functions if there are the stored only one or two amino acids. Now the frame shift mutations, it arises from the insertions or deletions of one or two base pairs. Since the codes consist of precise sequence of triplet codons, the additions of the deletions of fewer than 3 base pairs cause the reading frame to be shifted the all coded downstreams. Frame shift mutations usually act very deleterious, harmful, and yield the mutant phenotype. In addition, the frame shift mutations also stop codon and produce is shorter as well as different sequence. Now the biochemical mutations. Biochemical mutations are those which have the changes in the biochemical of the cells, and these mutations often inactive in a biosynthetic pathway. They frequently eliminate the capacity of the mutant to mutate and essential molecules. A strain being such a mutations, it is unable to grow on the medium lacking the molecules. A strains bearing the such mutations, it is unable to grow on the media which lack the molecules but grow when the molecule is provided. Such mutants are called the auxotrophs and they are said to be auxotropic for the molecules they cannot synthesize. If the wild-type strains from which the mutants arose of a chemoorganotroph able to grow on the lab and containing only the salts and a carbon source, it is called the prototroph. Another type of the biochemical mutant is called the resistant trophs, and auxotropic and the resistants are quite different. If the wild-type strains from which the mutant arose, it is a chemoorganotroph able to grow on a minimal medium and containing the only salts. Another type of biochemical mutant is called the resistant mutant, and these mutants have required resistance to some pathogens chemicals, antibiotics. And the oxotropics and resistant mutants are required and important for the microbial